For this next video clip, we're going to talk more about B cells and T cells and start to learn about their features and differences. All right, so lymphocytes. So your two main types of lymphocytes are B cells and T cells. And then as we move more into T cells, you're going to see that there are different versions of those that have different jobs. But okay, both B cells and T cells on their surfaces, and we're going to take a closer look at their surfaces shortly, they have receptors on their surfaces that bind to specific markers. And those markers are also called antigens. So we've talked about that already. Antigens are immunogens, molecules that stimulate these B cells or T cells to respond. So just keep that in mind. There are B cell receptors, there are T cell receptors, and these are molecules that are programmed to bind very specifically to some specific antigen, a molecule that you might find on a pathogen. Now there is a difference. The B cell receptors bind to soluble antigens. So what that means is if you've got a antigen molecule itself that's floating around in a fluid, or a soluble antigen could be a virus particle that has these molecules on its surface that are functioning as antigens. Or it could be a bacterial cell that has molecules on its surface that are functioning as antigens. So B cell receptors will attach to those things directly. T cell receptors, as we'll see, don't bind to antigens on a virus, antigens on a bacterial cell, or say a protein molecule that's just floating around into the lymph node. T cell receptors only bind pieces or fragments of digested antigens, and those digested antigens are displayed on the surfaces of phagocytic cells, so especially macrophages and dendritic cells, or as we'll see, other cells of the body can do this as well. I know that might not make sense yet, but it'll make more sense as we cover T cells in more detail. So T cells have to have antigens presented to them. So the antigen molecules of things that are foreign and invading the body actually have little pieces that get displayed on the surfaces of phagocytic cells, and they present those or show those to T cells. And that involves special cell surface proteins that we'll talk about in more detail. They're called MHC1 and MHC2. And down here, here's a diagram from your textbook which is showing a cell membrane. This is just a generic cell membrane from a human cell, and this little thing that kind of looks like a string of balloons up here, that is supposed to represent what we call a class 1 MHC protein. And so these are proteins that you have on all nucleated human cells. So any cell in your body that has a nucleus will display these class 1 MHC proteins on their surfaces. And the reason for that, and we'll talk more about the reasons as we get deeper into this particular chapter. All right, and then certain cell types certain types of white blood cells have another type of molecule called class 2 MHC, or MHC2. So this picture from your textbook, this thing that looks like a brown clamp sitting here in the membrane of this cell. So that's another protein that's involved in antigen presentation to T cells, but it's present only in the membranes of certain white blood cells, or white blood cell derivatives like macrophages and dendritic cells. And notice those cells will also have the class 1, because you have class 1 on any cell in your body that has a nucleus. And really, that's really excluding your red blood cells. So red blood cells don't have a nucleus. You learn about that when you take Biology 202. All right, and as we're going to see, B cells and T cells have very different jobs. So the binding of antigens to these surface receptors is what stimulates the B cells or the T cells to become activated. It stimulates them to take on this role of helping you fight off an infection. Stimulated B cells, as we'll see, go on to secrete antibodies from those little Y-shaped protein weapons we've talked about earlier. So we'll learn more about those. And then you have more than one type of T cell that have different jobs after they get stimulated. So, and the ones that we're going to focus in on are helper T cells and cytotoxic T cells. Helper T cells, as the name suggests, they go on to help stimulate your B cells, other T cells, and also phagocytes to carry out their actions. That's why they're called helpers. And some people consider these to be the grand conductors of your immune system. They're very, very important. People who have HIV infection and have developed AIDS, the reason they have AIDS, this immunodeficiency syndrome, is because, all right, I got interrupted there. So helper T cells get wiped out. The HIV virus, human immunodeficiency virus, kills helper T cells. And then the immune systems of your HIV/AIDS patients basically collapses. So they wind up getting all kinds of terrible infections, and that's what will ultimately kill them. So that just kind of emphasizes the importance of helper T cells in helping these other branches of your immune system do their jobs. And there's another type of T cell called a cytotoxic T cell, and these have very different jobs. They actually hunt for cells in your body that have intracellular pathogens. So mainly viruses, but there are also bacteria that go inside your cells. There are protozoans that can go inside your cells as well. There are yeast that can go inside your cells. And cytotoxic T cells identify cells in your body that are infected and kill them. And that's helping to limit the spread of those pathogens to other cells within the body. So they're basically assassins for cells in your body that are infected with some sort of intracellular microorganism. All right, so let's take a look at, this is a diagram from your textbook, which is showing you over here, this is supposed to represent a B cell surface, and this is supposed to represent a T cell surface. Okay, and a B cell surface, the main thing we're going to focus in on here are these Y-shaped molecules that you see. And those probably look like antibodies to you, if you remember the Y-shaped antibody molecules that we talked a little bit about back in chapter 12, and we'll be talking a whole lot more about here. And they really are antibody molecules, but when they are embedded, when they're on the surface of a B cell, they're called a B cell receptor. So that's a B cell receptor, that's a B cell receptor. And they're scattered around, so the surface of any B cell is going to be coated with these receptors. Also notice they're showing you the little brown clamps. Those are MHC2 markers, or class 2 MHC proteins. So B cells have those on the surface as well. And we'll learn more about why that is when we get to the video clip where we learn more about B cells. All right, now over here on a T cell membrane, right here in green, this is called a T cell receptor. So you notice you've got those scattered around the surface of the T cell. And so that's going to bind to an antigen. Same thing with the B cell receptors. Those two little ends of the Y there, those combine with very specific antigens. Same thing with the T cell, it just looks a little bit different. And then notice this T cell membrane over here has a CD4. That's a protein molecule on its surface. And you'll find that on your helper T cells. Cytotoxic T cells have something kind of similar called CD8, and we'll learn more about that as we go through the chapter in more detail. Now your textbook is showing a CD3, these little blue spikes. That's another kind of protein on the surfaces of T cells, and don't worry about that. That's getting into more detail than we need to cover for an introductory microbiology class. All right, now here's what's really interesting. So you generate many, 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 many different B cells and many, many, many different T cells with receptors that can bind to different antigens. Now for any one B cell, all of its receptors on the surface, and this is something you probably want to write down. For an individual B cell, all of its receptors are going to attach to or combine with the same antigen. Same thing for a T cell. You got an antigen binding receptor, the T cell receptor right there. All of those receptors you see on an individual T cell are going to bind to the same antigen. But you Now, obviously, not all antigens are the same. So what that means is that your immune system has to crank out billions of different B cells and billions of different T cells with receptors that can attach to almost any kind of antigen we encounter. So an individual B cell or T cell will bind to one antigen, but your immune system basically generates a library of B cells and T cells with slightly different receptors that can attach to different antigens that you might get exposed to. So there's great diversity in these B cell receptors and their T cell receptors. And that's important because you get exposed to all kinds of different microorganisms and other foreign things. And so if you need to stimulate B cell and T cell immunity, then you're going to need B cells and T cells with receptors that can bind to all of these different things that you get exposed to. Now, how in the world does that happen? So we just said that you generate B cells, many, many different B cells that can have receptors that bind to many, many different things. And the same thing with the T cells. And those receptors are proteins. So back in Chapter 8, we learned... Sorry, I just got interrupted again. We had a hot dog advertisement being broadcast here over the Huntsville, because hopefully you didn't hear any of that. But anyway, all right, so B cell receptors and T cell receptors are proteins. So back in Chapter 8, we learned that for any protein you make, you got to have a gene on your DNA that codes for that particular protein. But we just said that you're generating an almost unlimited supply of different B cell receptors and T cell receptors. So how in the world is that possible? We only have a limited amount of space on our DNA that can have genes that code for different proteins. So how in the world is it possible that we can generate so many different B cells and B cell receptors and T cell receptors? We simply don't have the space on our DNA to accommodate that. And so that kind of stumped scientists for a long time. But what was discovered is that there is some genetic shuffling that goes on for B cell receptor genes and T cell receptor genes. So you see, if this is a gene that is coding for, this is supposed to be a B cell receptor down here. So remember I said it's the tips of the receptor that actually attach to an antigen, like those two tips might attach to some molecule on the surface of influenza virus, for example. So the little colored portions there, imagine that up here, these are the genes on the DNA that code for the amino acids that are located here in the variable portions of the receptor. And so what happens is there's gene shuffling. So these little stretches of DNA undergo a shuffling process, and that scrambles the AGCT letter code there, which leads to the tips of different receptors having different amino acid sequences. Because if you shuffle the DNA up here, that's going to shuffle the messenger RNA transcripts. That's going to shuffle the amino acid sequences in the proteins, which is pretty cool. And same thing happens with the T cell receptors. You've got the same kind of shuffling that goes on within the DNA that allows for that diversity to take place. Your textbook has a little bit more of an explanation on that, but just wanted to point that out. You know, how is it genetically possible that we could have all these gazillions of different B cell receptors and T cell receptors available to bind to different antigens that we get exposed to? And incidentally, the people who discovered that actually won the Nobel Prize for their work, because it was such an important discovery. All right, so when if you've got an individual B cell, an antigen comes in and attaches to a receptor on a B cell, that B cell will, now it gets a little bit more complicated, and we'll learn about that, but that B cell, when it becomes stimulated, it will divide over and over and over again. And same thing for T cells. When an individual T cell gets stimulated, it's going to divide over and over and over again, and we'll learn about that as we get into a little bit deeper into the chapter. So an individual, there's a term clone here, and when we're thinking about B cells and T cells, an individual B cell clone is a single B cell. A T cell clone is a single T cell. A B cell clone attaches to its receptors will all bind to the same antigen. Same thing with a T cell. A T cell clone, its receptors will all bind to a particular antigen. So we just mentioned how through this genetic shuffling that takes place, we wind up generating many B cell clones and T cell clones that can combine with or attach to different antigens that we get exposed to. Now, interestingly, take a look at this diagram over here. So they're showing you've got a lymphocyte stem cell that generates a bunch of B cells or T cell clones, and they have receptors that will bind to different antigens. So with all that genetic shuffling that's going on, what prevents you from having B cell receptors or T cell receptors that will attach to your own antigens, your own surface molecules? And incidentally, that does happen. However, before B cells get out of the bone marrow, or before, remember, T cells go to the thymus to mature, before T cells leave the thymus, they get tested to see whether their receptors attach to any of your self molecules. And that's what they're showing you over here. So here's one of your cells with self molecules. Here's another one over here. And here we've got a lymphocyte whose receptor attaches to a self molecule. Same thing over here. When that happens, those lymphocytes are supposed to die. That's called clonal deletion. So lymphocyte clones that attach to your own antigens, molecules on the surface, are supposed to die. That way you don't respond to your own antigens, your own molecules. Now, sometimes this clonal deletion does not work, and that's where you can develop a type of autoimmune disease where your immune system is attacking your own cells or tissues, and we'll learn more about that when we get into chapter 14. All right, and sometimes this is called B cell education and T cell education, so they're getting tested before they're actually released and they go into the blood and go to the lymph nodes and go to the spleen, go to the tonsils. Any B or T cells that can attach to your own antigen molecules are supposed to die. So you wind up with a repertoire, a group, a whole bunch, like a library of lymphocyte clones, individual B cells and T cells, that have unique receptors, and those wind up going to your tonsils, going to the spleen, going to the lymph nodes, and so forth. And they basically hang out there. And when antigen molecules of some sort of foreign invader, like a bacterium or a virus, attach to those receptors, that individual clone divides over and over again and creates a big population. That's called clonal expansion. So be sure you're familiar with the term clone, what clonal deletion means, and then also what clonal expansion is referring to. Now, you've felt clonal expansion before, because if you've ever had a sore throat, you reach up here and you start feeling around your cervical lymph nodes, they get swollen, and that is because you have B and T cells in those lymph nodes that encountered their antigens that they're programmed to respond to, their receptors attached to the antigens, and that caused them to divide over and over again. They underwent clonal expansion, and you're creating an army of those B and T cells that are available to help you fight off that invading microorganism. All right, next video clip, we're going to talk a little bit more about what antigens are, what does that mean, and what makes something a good antigen. And then also we're going to learn more about T cells. We're going to do T cells first, and then later we'll do B cells. So how do these T cells, with their little receptors on the surface, get stimulated? And remember, we said that they have to have antigens presented to them. Little fragments of antigens have to be shown to them by phagocytes. And so we'll see how all of that works in the third video lecture for chapter 13.